that so, so that Grace and the folks who aren't here can hear. So today we'll go through some mechanisms, and there will be 2 videos posted, one on enzyme regulation and the other on basics of carbohydrate structure. And then the class, when we get back, we're going to go through lots of practice problems and then talk about applications or structure or functions, and regulation of carbohydrates. So, all right, so we had gone through just the overview of the mechanism of chymotrypsin, okay? And I mentioned that there were 2 phases, right? So what we're going to start with is going through those phases in detail, and particularly pointing out features of the active site and the catalytic triad that are important for the mechanism. Okay, so to remind you that chymotrypsin is a protease, okay? So its function is to cleave peptide bonds and again, I had mentioned last class that there were a number of protease enzymes, and they all have different specificities. So for chymotrypsin, it cleaves adjacent to aromatic residues, okay? So if you'll notice, again, in this overview figure, what you'll see is you see this pocket that has a phenyl ring in it. Okay, that's a phenylalanine, and so this is the region that that's responsible for specificity. We're going to look at that in more detail later. Hi, Christopher. All right. All right, and there are 2 phases, okay? The first phase, okay, is where we form the covalent intermediate, okay? So this is covalent catalysis. And the bond that's going to be cleaved is cleaved, okay? So cleavage of, when we use the word sessile, that word comes from the same root as scissors, okay? Same root as scissors. And it's a common term used in biochemistry when there's a bond that's going to be cut, okay? So if we say the sessile bond, it's the bond that's going to be cut. Okay, so let's go through the 3 steps that are in this phase, okay? All right. So in step 1, okay, you get binding into the active site. Okay, so if we're looking for, our substrate, here's the substrate that's coming in and it's binding. Okay, and so now here's the substrate right here. Okay, and again, so that we can show it in 2 dimensions. Okay, the sessile bond, which I'm hopefully going to highlight in pink. Let me just see. So it's going to be the peptide bond right here. Okay, so it's our peptide bond. Okay, so it's shown very much stretched over here. Oops. Hello. There we go. It's shown very much stretched just so that we can show things in 2 dimensions. Okay. So when we get binding into the active site, you're going to be getting small structural rearrangements, okay, that bring 2 of our important active site residues into closer contact with one another. Okay, so this is our His 57, okay, and our serine 195, which is over here. Okay. So you'll notice that we get our histidine, which started out in its neutral state, okay, is going to actually deprotonate the serine, okay? So the serine hydroxyl is going to lose a proton to the histidine, okay? So we've got the serine, 195, acts as a general acid. Histidine, bless you, 57, acts as a general base. And then once you have, okay, the serine deprotonated, okay, what does that leave you? So your serine before has a hydroxyl. So this is before and after. Your serine is an oxyanion, right? So that is going to make a nucleophilic attack on the carbonyl carbon, okay? So the serine oxygen makes a nucleophilic attack, okay? And over again, we've talked about carbonyls, how that bond is polarized, and that carbon actually has a partial positive charge, right? So just a reminder, right? Carbonyl is polarized. All right, so that's the step where the covalent intermediate, where your enzyme is connected to part of your substrate, is formed, okay? So this right here, covalent intermediate between enzyme and substrate. And so what we get, okay, and if we look at it in the next slide, right? So here's our serine, this bond right there, okay, between the carbonyl carbon and the serine. This bond right here is the bond that formed between the enzyme active site residue and the substrate. Okay? All right, so you'll often hear when you look at enzyme mechanisms about tetrahedral intermediates forming, okay? And so if you look here, right? Oops. That wasn't what I wanted. That wasn't what I wanted. I wanted this. There we go. We're using the term tetrahedral just like we use any, talk about geometry when you first learn geometry in Gen Chem, right? When you have carbon attached to four things, the angles around it are what, like 109 or something? And so you've got four things coming off, four covalent bonds to your carbon. So the physical geometry, the spatial arrangement around that is tetrahedral. So that's all that we mean by that, okay? All right. Whoops. Come on. There we go. All right. So our carbon is connected to four things. And that was the carbonyl, right? Okay. So, but the next thing that happens, okay, so first we have the serine making the nucleophilic attack. But now we have that histidine that's protonated, okay, and that histidine, okay, is now having acted like a general base, it's going to act like a general acid again, and it's going to donate a proton, okay? And it's going to donate that proton, to the amino group of the substrate. Okay, so that's this amino group here. Okay, so oops, it's this amino group here that's on kind of the amino side of the scissile bond. Remember, this is our scissile bond. There's the carboxy side, there's the amino side. So we have this bond between the serine and the carbon, and then that makes this bond, right, susceptible to cleavage, okay, when that amino gets deprotonated, okay? So the important part of this, okay, is two things. One, we get peptide bond cleavage, okay? So we get peptide bond cleavage. That's what this thing is right here that I put the check mark next to, okay? And then the second thing that happens is the carboxy terminal fragment gets released as the first product, okay? Now, you might be thinking, wait a minute, we just protonated the amino side of the scissile bond. Why is it talking about the carboxy terminal fragment getting released, okay? So let's just talk about where the amino and carboxyl termini of the peptide that's being cleaved are, okay? All right, so if this right here, okay, if this part right here is going to be the carboxy terminus, the amino terminus of the two fragments, that means that this side over here was the carboxy terminus, right? So this is the C terminus, which means over here, okay, when we cut this bond, this is going to be the carboxy terminal, and that means that this end over here, whatever there is over here, is going to be the amino terminal. So this here is the N terminals, okay? So when we say the carboxy terminal fragment is released, that means this part. Okay, so the part that is not covalently bound to the enzyme is released, okay? So this is the half of the peptide that is not bound covalently to the enzyme. All right. So what does that leave us with? All right, well, what that leaves us with is this N-terminal fragment covalently bound. So let's do this. All right, so the thing that we're left with is the fragment bound to the enzyme in the active site, right? Now, what that also leaves us with is an oxyanion that develops, and you see it highlighted in orange here. Here's the minus sign there. So there's our oxyanion, okay? That has a negative charge, right? So this whole process is going to be favored if we can stabilize that negative charge, right? So that's where we have what's called the oxyanion hole. Okay? And you'll notice, okay, that you'll see a couple of other amino acids, okay, that are forming, what you see over here, hydrogen bonds with that oxyanion hole. So remember that the hydrogen that gets involved in a hydrogen bond it has a partial positive charge. So think of these hydrogens as partial positive charges, okay, that are going to stabilize this negative charge there by forming a hydrogen bond with them. So the question is, where do those residues come from, okay? And where do those hydrogens come from? And the hydrogens, and this is kind of unusual, right? The hydrogens come from the backbone amino groups. So remember serine 195, is it is the residue that forms the covalent intermediate. Okay, so if we look at, here's our alpha carbon, here's our side chain atoms. So I'm going to label this as the C alpha, okay? And then in the enzyme, right, that carbon alpha is going to be attached to a carboxyl on one side, and then like the amide nitrogen on the other. So this is the amino, or the amide nitrogen, that belongs to serine 195. So, and then the next amino acid that's there is that contributes an amide nitrogen is glycine 193. Okay, so that's, let me make that better. There we go, glycine 193. Okay. So in terms of the identity of this amino acid at position 193, okay, it doesn't matter so much what the side chain is, because the backbone is playing the important role in the stabilization of this intermediate, okay? But it is important in terms of the fold of the protein. So these backbone atoms have to be in the right position in the oxyanion hole, right? So let's do this. This right here is my oxyanion hole. Okay. So can I replace this glycine with something else? Perhaps, right? But it has to be something that's not going to disrupt the structure, but with its side chain. It has to allow the chain to fold so this winds up in the right place. So maybe replacing it with an alanine would be okay, but like a tryptophan wouldn't, or a proline wouldn't, right? Because that would, particularly the proline, because the proline's gonna affect the backbone a lot. Okay. Okay. So all of that is phase one. Okay. So now we have a problem. We have two problems. One, the other half of our substrate, our product, is stuck to the enzyme. And two, we need to regenerate the active site of the enzyme. Okay? So we've done a little bit of that already, right? So if we look at the histidine started out deprotonated, it got protonated, and then it donated that proton back. So our histidine, okay, it's in its correct protonation state to do its job over again. Okay. The aspartic acid, remember, the only reason that this is there is its job is to position the histidine 57 in the active site, just so that it is like pointing in the direction it needs to go. So it forms a very important hydrogen bond just to hold that thing in place. So its job is to H bond with histidine 57 to keep it properly positioned. Okay. All right. So now we're going to talk about phase two, okay, where we solve both of our problems, right? We need our problem number one, we need to release the amino terminal end of our substrate. And problem number two is we need to regenerate the active site. Okay. And the key player in this is the fact that, that we need a water molecule to come into the active site. Okay. So let's go back. Key player is a water molecule. All right, so zooming in. All right, so when last we left our active site, okay, we've got that we've lost the carboxy terminal of the peptide. We have the amino terminal stuck in the active site with that oxyanion stabilized by hydrogen bonds with backbone residues in the oxyanion hole. Okay. So now what we need is our water molecule to come in. So here's the water molecule, okay, that enters the active site. And that water molecule, okay, is going to interact with that histidine. We thought that histidine was all regenerated in its final, in its ready for next catalytic cycle form, okay? But when the water comes in and we still have that other half of the peptide there, okay, the histidine again is going to act as a general base, and it's going to remove a proton from water, okay? Okay, so now instead of the serine acting as a nucleophile, okay, this time the water molecule is going to be the nucleophile that attacks a carbonyl, okay? And where is the carbonyl carbon? The carbonyl carbon, okay, is the carbonyl, okay, that formed, okay, between the serine residue and our N-terminal fragment, okay? So we see the same thing happening all over again. This is going to break down, okay, the acyl enzyme intermediate, okay? So we're going to get, so what we're going to get is, here we go. We're going to get this bond being broken, okay? And the water, okay, is going to essentially be added, okay, to that bond. So here's our water. Half of it winds up with the N-terminal fragment, okay, making the carboxylic acid C-terminus of that N-terminal fragment, okay? And the hydrogen from that water, okay, winds up, protonating that histidine residue. So, oh my gosh, now our histidine is protonated again, and we got to get rid of that proton, okay? So, what's going to happen? Okay, is the way we're going to regenerate everyone in the way we want, right? So at this point, we've got a protonated His 57 and a deprotonated serine 195, which is exactly the opposite of what we want to have an active site that's ready for another round of catalysis, right? But that's what you have kind of at the end of step 4 here, right? So step 4, this happens, we get this as a result, okay? So the next thing that's going to happen, right, may seem obvious, is that histidine is just going to give the serine back that proton, okay? Although it isn't the original proton that the serine had, right? The original proton that was on the serine wound up leaving as a proton on the amino terminal end of the C-terminal fragment. And the proton that eventually winds up on the serine, okay, it comes from water, passed to the histidine, then passed to the serine, okay? So we get product 2 leaving, okay, so that leaves, right? And then we have the original state that we started with, okay, where we have a deprotonated histidine 157, and serine 195 is protonated. And the substrate has left the active site, so we have nothing left in here in terms of our extra atoms, and the structure is going to relax, okay, in such a way that this histidine is not going to take that proton back from the serine. Is anybody wondering how we know what proton goes where and what proton comes from where? Or no? You're just going to take my word for it? Yeah, you can take my word for it. That's okay. But I will take this moment to point out that the way we figure these things out, okay, if we want to, figure out this whole mechanism here, okay, is a combination of predicting, okay, what you would get using a conceptual model that you connect to things you can measure in the lab kinetically, okay, using mutations to change different residues and see, if we tweak this, this residue, maybe make its pKa higher, or if we, change it so that this is a little bit, the side chain's a little bit longer, maybe instead of an aspartic acid, we use a glutamic acid, so we have it further projecting into the active site because it has an extra methyl group. Okay, so it's a combination of those things and isotopic labeling. Okay, so what we can do, right, if we want to say investigate, okay, we know we've done crystallography, we know the structure of this thing, we know that there's enough space for a water molecule to enter. We've got a transition state analog that kind of locks us in that, maybe that acyl enzyme intermediate, because maybe we've substituted an atom in the substrate with something that maybe like a fluorine atom or something, so that we can get the original cleavage, but then we can't get maybe release of the substrate, right? So we can have labeled substrates, or maybe labeled transition analogs, state analogs, and we can do things like using heavy water. Okay, so you can put your system into a solution that is made up of deuterated water, okay, and then we can look for where the deuterium wound up, okay, or we can use tritium labeling on the hydrogens, okay, and look for where those isotopes wound up. So that's how we can tell definitively, like, no, this hydrogen came from this water molecule. This oxygen came from this water molecule. And it's a really common way of being able to follow individual atoms when we're looking at these complex biological systems where you've got thousands and thousands of atoms, and you want to know, something very, very specific, like, okay, where does this hydrogen come from? Okay, we can use isotopes to answer that question. Okay. One of the best talks that I ever heard, and this is not going to mean much to you guys, but way back when, okay, in college basketball, there was I think they were called like the Fab Five. They were University of Michigan, I think, basketball team. What's that? Did you say gross? Yeah. So the University of Michigan had this, championship-winning basketball team that had these 5 players that were just worked perfectly as a team. And there was I went to a talk. It was a special biochemistry symposium at an ACS meeting. And this guy who's a professor at Penn State called Squire Booker, he was talking about work they did elucidating an enzyme mechanism literally by following an individual proton where it went. And it just so happened that there were like 5 amino acids that were involved in this mechanism, or this proton went to 5 positions. And his whole talk was this like analogy with these 5 residues being like these 5 basketball players. And it was so cool. And I wish I had like remembered the talk better. But anyway, so that was really the first time I had seen, I had actually listened to a talk about like how they figure these things out, and it was super cool. All right, so we've talked about general acid, general base catalysis, a covalent enzyme intermediate, covalent catalysis, and we've talked about the role of the oxyanion hull. So the last thing that we have to talk about with regards to chymotrypsin and serine proteases in general. So remember that serine proteases are a class of enzymes, okay, that all use these 3 residues for catalysis: the aspartic acid, the histidine, the serine, okay? But they do not act on the same substrate. And so let's take a minute to talk about this thing right here called the substrate specificity pocket. Okay? So this is going to be the portion of the active site that differs between different serine proteases. So if we look at it, oops, and I'm going to move this. Let's see if we can move this over here. There we go. Okay. So when we talk about active sites, we will often talk about specificity pockets, okay? And we'll define the regions of the active site that are responsible for the initial binding of the substrate, that first step, right, in our conceptual picture of what enzymes do, okay? And so the terminology that we use, right, is we use for sites that are on the peptide, okay, those we label P for peptide. Not very shocking, right? So potential sites of interaction of substrate with enzyme, those are called P on the substrate, okay? And then the partner site, the partner that it's going to interact with on the enzyme, are labeled S for subsite, okay? So this is the binding partner of the P residues or atoms, okay? All right, and so again, we're talking proteases specifically, okay? And then remember that we use this term cis-aryl bond to refer to our cleavage site, okay? So this is our cis-aryl bond. And the cis-aryl bond defines our numbering, okay? So we number, the higher your number, the farther you are from the cis-aryl bond, okay? So the higher the number, farther from cis-aryl bond. All right. So if you're looking, if you're looking in the literature comparing proteases, whoops, this is what you'll see in terms of terminology. Okay. So what we're going to do is we're going to look at 3 examples. We're going to compare chymotrypsin to 2 other serine proteases that have different specificities, okay? And we're going to see how the sites compare, okay? All right, so all 3 of these are serine proteases, okay? So we've got serine proteases, and they all use similar catalytic triads, okay, the same catalytic triad, aspartic acid, histidine, serine, okay? So this slide is not showing you, okay, where the catalysis happens. We're zoomed in to the active site, okay? So these views are only of the specificity pocket. So that's the first thing to note, okay? All right. So the three, chymotrypsin, okay, binds adjacent to large hydrophobic side chains, okay? So chymotrypsin, large hydrophobic side chains. Trypsin, okay, cleaves on the carboxyl end of large positively charged side chains. Okay? And then elastase, okay, cleaves on the carboxy end of small nonpolar side chains. Okay? So we've got aromatic hydrophobic residues, we've got residues with a positive charge, and then we have small residues. So that's the difference in specificity in terms of what's going to bind in the active site. So how do we choose between these? Okay, so when in chymotrypsin, okay, we have enough, first of all, we have enough space, okay, to accommodate that residue, okay? So the enough space part is the glycines. Glycines are very small, right? They don't have anything more than a hydrogen on their side chain. So if your glycines are lining that specificity pocket, there's a lot of room in there, okay? So we've got large, roomy pocket. And then the other thing that we have is at the base of the pocket, okay, we have a serine residue, okay? And that serine residue helps to bind aromatic side chains, okay, like the one that's shown in this image. What's the side chain that's shown binding in that image? What do you have? So we have a phenyl ring, and what's on the end of it? Hydroxyl. What's that amino acid side chain? Close. Say first letter correct. Tyrosine. Yeah, so this is a tyrosine. Okay. Ooh, that doesn't show up very well, does it? Let's change that to, there we go. All right. So we've got a tyrosine here. This is a hydroxyl, okay? And serine, remember, has a hydroxyl. So when a tyrosine comes into that pocket, okay, there is something for it to hydrogen bond to. Now let's say that hydroxyl group, okay, so if a tyrosine enters this pocket, this hydroxyl group can hydrogen bond to the serine hydroxyl, okay? What about if there's no hydroxyl here? What's that residue? If all we have is a phenyl group on our side chain, we have phenylalanine, right? Okay, phenylalanine still going to be able to enter the pocket, right? It's going to, these are hydrophobic residues, okay, the glycines. So you'll be getting, you know, whatever van der Waals contacts you have, any pi-pi interactions that might stabilize the phenyl portion. But it's not going to care so much about the serine being there, right? So that's not going to prevent it from binding. So that's how this binding site can have, you know, two different amino acid residues projecting into it and still be happy, okay? All right, so trypsin, right? It's going to be large positively charged, so arginine and lysine. Those are the guys that are going to come in here, okay? Which one is shown here? Is it arginine or lysine? It's arginine, yeah, because lysine just has the amino group, right? So this guy right here is arginine, okay. And that's positively charged. So large, we still need to have a big binding pocket, right, with slightly hydrophobic things that can accommodate the long chain that's hydrophobic itself, but then we have this charged group at the end. So we're putting a negative charge at the bottom of that pocket, and aspartic acid is going to enable a salt bridge to form between these two atoms. What if we put a glutamic acid there? What do you think the consequence of that would be on binding? So how would a glutamic acid change this? It would still be negative, right? So you would still have this favorable interaction. Is a glutamic acid longer or shorter than an aspartic acid? So the aspartic acid is the shorter one. And the way I remember it is they go in alphabetical order. So aspartic is shorter, glutamic is longer, and it's further in the alphabet. That's my super scientific way of remembering it, right? So if we have something that projects a little further into this, you could imagine that you'd still be able to bind, but maybe you would bind a little less well, right? So in which case, when we get to doing examples about looking at those kinetic factors, you would expect to see the Km value increase, because you'd need more substrate to get as much binding. And then the very last one, right, is elastase, where we want to only allow small things in and small hydrophobic things. So we just put residues that are going to fill up that space, okay? So avaline, threonine and serine can hydrogen bond with one another because they both have the hydroxyls. That's going to kind of keep them neutral, right? And so this is going to be, we're not going to get too much bulk in there, but we're going to fill up the space with atoms so that all we get is this little one. Okay, so that's the way that just changing 3 residues in that specificity pocket enable you to pick what you want. Okay, so it's just like changing the shape of the puzzle. All right. Thank you for coming in, and have a great Thanksgiving.